like to welcome everyone here to the University of Saskatchewan, and this is the distinguished lecture series that is organized by the Global Institute for Water Security. It's part of the Global Water Futures program, and also contributes to a class in the School of Environment and Sustainability, both in Saskatoon and in Beijing. So I want to welcome those of you early morning in Beijing who are online for this lecture. This week we're very honored to have Professor Martyn Tranter speaking to us from the University of Bristol in England. Martyn is director of the Bristol Glaciology Centre, and in fact he has a long time connection to Saskatoon, which I'll go into in a bit. But he's been at Bristol University for about 27 years, previously at the University of Southampton, and a PhD from the University of East Anglia. His research has focused initially on snow chemistry and then increasingly in glaciology, and he was a very important member of a group that developed the concept of snow ecology, that microorganisms in particular were extremely important to the snow and ice environment. And he's taken this further by really developing the field of glacier ecology, which is not something you normally think would be a field, but he's going to show you that glaciers are alive in many ways, and you'll never eat snow and ice again that after this lecture. He's, despite all these collaborations, he's, and because of them, he's become very prominent. He's received the Polar Medal from the Queen. He's led a multitude of very large studies, including a wonderfully named current one called Deep Purple. It's funded by the European Union, and so as long as Brexit doesn't happen, it's going to be great. For them, but and he's also been a wonderful collaborator to those of us in Saskatoon over the years. Back in the 1990s on snow chemistry and ecology with studies up in the boreal forest, and then also analyzing data that came out of northwestern Canada in the Arctic, and then through collaborations such as the Ecology Working Group. This is not his first seminar in this room, and in fact, if you go back to the early 1990s, he was a relatively young scientist brought in here by another young scientist at that time for the what was called the seminar series of what was then the National Hydrology Research Institute. And so despite that experience, we've invited him back, and his chance for redemption in this case. But also it's wonderful to look at the progression of a scientist's career over a multi-decadal period. So Martyn, thank you for coming, and turn it over to you.
Oh, thank you very much, John. It's an honor and a privilege to be here, and I've just had the best of times. It's been a pleasure and a privilege again to meet with so many young scientists with energy and enthusiasm doing exciting work. And boy, you know, you've got a fan for a long, long time here. You know, and I want to come back, and I want to collaborate more with some of you in particular. So what I want to do today is maybe talk about something that you don't do normally in your day jobs. It's to think about the darkening of the Greenland Ice Sheet and what's causing that darkening. And I hope I'll convince you that biology has a big part to play in this darkening. And I'm going to pay my dues to Jason Box and Marek Snow, who started the work off on the Dark Snow project. I want to talk you through some work we did, or we're just finishing on the Black and Bloom project, which was a NERC large grant, multi-institutional. We hired some fantastic young scientists who I think are going to make an even bigger splash in the pool in years to come. And then I thought I was retiring in a year's time, and we've got an ERC synergy grant. Alex Anisho, Liana Benning from Aarhus and GFZ Potsdam. And the irony here is it looks like I'm going to be on a 6-month wind down into retirement, but I hope doing some more good research as we're on the way. So I take the hit. Any rubbish here is all down to me. The real stars of the presentation are the 4 Black and Bloom postdocs, Joe Cook, Janine McCutcheon, Andrew Tedstone and Chris Williamson, Our PhD students are in the next row, and the parasitic PIs are the final row. And then my acknowledgement to Jason Box and Marek Stibble for letting us in on the game in 2014. So, what's the why the interest in biological darkening of the Greenland Ice Sheet? Well, in the UK, we've always had a big interest in the Arctic, and the societal need for the work is the prediction of future sea level rise as a result of the progressive melting and potential collapse of the ice sheet. And for example, at the moment, Greenland's contributing about 0.8 millimeters per year to sea level rise, and a tipping point is predicted at the moment when the climate warms by another degree C. And then, a bit like Pato Glacier that some of you have been talking about, there won't be any accumulation, there won't be a snow line, the whole ice sheet will be melting. And when this happens, over several hundreds of years, hopefully, Greenland can contribute up to a 7.3 meter rise in sea level. So whether the melt is going to accelerate in the coming decades, whether this tipping point is hit sooner or later, those are pressing needs, research needs. And one of the big issues for determining how much melt there is and how fast the melt is going to happen is the albedo change in the surface. So much of the melt on the ice sheet occurs down the southwest margin and the southern tip of the ice sheet. And over the last 20 years, the amount of melt has been increasing, and it's the biggest mass loss term from the ice sheet now. And over the last 20 years, and the last 10 years in particular, the albedo has been decreasing down the western margin, and you can see that this albedo change is by 0.1 and more of an albedo unit. An albedo scale is only from 0 to 1, so these changes are quite large. This is some work that Andrew Tedstone put together, and it shows you year by year variations in the extent and the intensity of the development of the dark zone. And you can see that the dark zone doesn't sort of get dark year on year. There are some years, big melt years, when it gets very dark, and there are other colder, later spring years where it doesn't darken very much at all. And the question is, what's controlling the albedo change? And we set ourselves in the Black and Bloom project the idea of somehow sampling all around the western margin of the ice sheet, so that we could learn something about albedo darkening processes. When we started looking at where to sample, we realized that the dark zone's just not uniformly dark. Even on the length scales of 30 kilometers or so, there's quite a lot of variability in the darkening. We go down again to the 1 kilometer scale, and you can see that there's a heck of a lot of heterogeneity in the darkness. On a 10 meter scale, the same sort of thing. Heterogeneity is everywhere, and even down to the 10 centimeter scale. There are patches of light ice, dark ice. This is over some cryoconite, and you can see how black that is. And we were somewhat alarmed by how much heterogeneity there was in the darkening of the surface. So we set ourselves quite a tough task, somehow explaining all the darkening of the ice sheet at all the various different scales, And you can see, I'll give you one of the conclusions that we've only barely scratched the surface. And where we came into the game was that the processes impacting on the darkness of the surface were thought to be windblown dust, be it from the tundra or longer range transport of dust. They were felt to be the melt out of dustier bands of Holocene ice in the ice sheet. There was a feeling that there was meltwater being ponded in the surface somehow. Of course, there was cryoconite holes. These little circular holes filled with dark sediment. But one of the big things that was felt to be darkening the ice sheet was black carbon blown from tundra fires. And Athabasca Glacier, I've been learning about, is covered in soot at the moment, and that seems to be that you would imagine that the darkening could have something to do with the with soot, enhanced soot deposition. Now, as far as the biology was concerned, when we came into the game in 2014, 2015, the only biology was thought to be in the cryoconite holes, and I love these things. I worked on them in Antarctica. They're goldfish bowls with sand at the bottom, and in the sand, tons of cyanobacteria grow. So this is a microorganism called nostoc. It's so robust, it can fix its own nitrogen if it's nitrogen limited. They're fantastic model systems, but typically they might only cover a few to maybe 8% of the ice surface, and they wouldn't be pervasively darkening the ice surface. So the only biology that people felt was inhabiting the surface of the ice sheet was cryoconite. And so what would cause the pervasive darkening of the ice sheet? Well, Jason Box did a great job for the discipline in suggesting that it was soot from wildfires. He got on the front of a Time magazine, showing that sooty ice melted faster than ordinary ice. A real charismatic figure who got enhanced melting of Greenland on the map. And I tip my hat to him. He's been so helpful. Great guy. But the thing is that you would normally expect this to be clean ice, and when you sample the surface ice in the dark zone, it really is mucky. If you melt it and let it settle out in a Falcon tube, you get this mush at the bottom that looks like soot. But if you look at the soot under the microscope, you'll see that they're purple pigmented glacier algae. Right? It's bonkers. And the other kind of odd thing is that where you find lots of algae, you find lots of particles. And it looks like you get a double whammy, that where the glacier algae grow, they also trap other light-absorbing impurities, other mineral dusts, and they also trap soot. So the bonkers thing is what you might otherwise think of is a dark sooty surface. As soon as you get your eye in, you can't find clean ice in the white ice at the top here, but everywhere else you can see darkness. Here are some tents, and this is the Dark Snow Camp in 2014. It's about 30 kilometers from Kangerlussuaq, which is the airport, Sonderstromfjord, old US Air Force base. And the field craft, the field site, Jason and who works at Geos in Copenhagen, and Marek Stibble now at Charles University in Prague, they kind of set us up to do the Black and Broom project. And here's some very, very provisional data from Marek. that we were able to use in the NERC show and tell grant pitch. And Marek very kindly let us use these data. So look, here's the albedo of the surface ice, going from 0.6 down to 0.1. Here is a log scale, cells per mil. And you can see that surface ice easily gets down to an albedo of 0.3. And indeed, some of the ice, particularly where there's dispersed cryoconite, can drop down to 0.1. So curiously, my remote sensing colleague John Bamber said, you know, that's as dark as the oceans, you know, and they're the darkest natural environment on the Earth's surface. I have to believe him, and I'm parroting what he said. So it looks like more algae, lower albedo. And the question we set in Black and Bloom is, well, what controls how the algae grow, and can we learn how they grow, how they darken the ice surface, so that we can produce parameterizations of albedo that are presently not used in ice sheet melt models. And as soon as we started, you know, not the media. You know, we're not as media savvy as you all are here, but we went straight into science, you know, the big alarms, meltdown. And we went from a situation, 2016, where honestly, I went to Gothenburg to talk to some ice sheet modelers, and I got laughed out of the room because they said, don't be stupid, biology has no impact on albedo. So the older scientists were very, very anti. And the younger scientists were, whoa, you know, we like this, you know, we believe it. So I got, you know, we got laughed out to court in 2016, and then by 2019, you know, it's a statement of fact that there are glacier algae growing on the ice sheet surface, and they are darkening the ice sheet surface. And this comes from the BBC News, 2019, 3rd of September. Forthcoming IPCC reports include sections on the need for an understanding of albedo decline in warming worlds. And where we're going to get to is that Deep Purple will try to deliver on what Black and Bloom started. So the idea to do this work, it's quite labor intensive. It's the scientific equivalent of having a heroin problem, in that it's hugely, hugely expensive, because everywhere you go, you've got to go by helicopter, and the helicopter is going to cost you about £7,000 sterling, so $10,000 an hour to fly. And then when you get dropped off, there's only so much you can do on the ice surface. And then to move around the ice, you need to hire the helicopter back in, and it's $10,000 an hour to do any work. The data is really, really hard won. So you might wonder what this lonely person is doing on the ice sheet, and at the same time, you know, the ice sheet surface dark. It's not grey. It's dark. There's stuff growing in the ice surface. And they are looking at an incoming storm. Now, my wife is a weather forecaster, and I took it upon myself to every day phone in what the current weather forecast for the area on the ice sheet where they were working was, and I missed the fact that there was a huge storm coming through. And so my poor team were in the middle of a gale, trying to batten down the tents, batten down the main work tent, and this thing is allegedly pretty indestructible, and got totally trashed. So You need a lot of contingency in your budget to do this type of work, and you need a heck of a lot of field craft to be able to undertake it. And hopefully, over the years, we've developed all those things. So, when you look at a dusty ice surface, what you think is dust is almost certainly some kind of pigmented glacier algae that's growing wherever there's life, wherever there's water, there's life. Ice sheet surfaces are no different. And why does this pigmentation arise? Well, the pigmentation is there to prevent the chloroplasts in the glacier algae being fried by the UV radiation. So over time, the pigments grow. Different types of pigments grow depending on the nutrient availability and the light intensity. But if you will, the hot sun-tanned algae are melting the ice sheets. So again, we put this paper out in 2012. I was talking to some of the postdocs earlier about being persistent. We didn't get the first grant. We didn't get the grant the first time, and we had to put it in a second time, just like the NSF system, before we got funded. And our central hypothesis was that these glacier algae, they grow pervasively in the rotten ice surface, which is otherwise known as the weathering crust. They're pigmented to protect themselves against UV radiation. And so it follows that as the microbes grow, they must change the surface albedo, and they must then increase the amount of melt that occurs on the surface. So we started the project off. We got 4 really bright postdocs, and the first thing they tell me is look, the main change in albedo is when the snow cover melts away and reveals the underlying ice. That's the principal change in albedo. And the snow might be running at an albedo of 0.9, 0.8. The ice melting out underneath is, at best, it's got an albedo of 0.6. Oftentimes, it's got an albedo of 0.5. And the biggest change in albedo is due to variations in the physical properties of ice and the overlying snow, without a doubt. And then the bioalbedo is a secondary effect, and it occurs largely in the visible part of the spectrum that our eyes can see. So what we wanted to do was to disentangle the impact of the microbes from other changes in the other physical and chemical properties of the surface, and to try to understand what the glacier algal effect was. Living up on the ice sheet surface is tough. You're camping on ice. The weather's very variable. It's fantastic environment, but 24 hours daylight, you're on the go all the time, and you can't stay up there too long and work to your maximum potential. And a way to understand how the albedo might change over a melt season is to do geographers classically do time for space substitution. So you fly up to the snow line, and this is, if you will, this is your pre-melt condition, and you can do a bunch of sampling, and then you fly over towards the ice sheet margin, and you get then an idea of how the algal blooms start growing over time. One of the things that sort of grabbed my attention the first time I was up there was that even though there's only there's still a light snow cover here, but it seemed to me that very quickly the ice underneath was already dark and was starting to grow the glacier algae, just like you find, for example, in the blooms that occur under sea ice, just like you find the algae that on the cyanobacteria that grow in cryoconite holes. So it seems to me that the algae start blooming just as the snow cover is starting to melt and to disappear. We stayed at this site all through the kind of July, August, September. So in the first part of the season, we did the if you will, the easier part of the year, the height of the melt season, and then the freeze up. And in the second field season, we did the harder part of the year, which is being put into camp on snow, and then have the snow melt around you to reveal the ice surface underneath. And so we got it took us 2 seasons to undertake our full cycle of work. All the time, I'm showing you photographs of ice surfaces, and all the time, I hope you can see that there are the glacier algae are growing in a patchy manner, and all the time, I hope you can see that the surface is dark. And microbiologists, they love to sample their algae, stick them in bottles, and then torture them by inflicting bright solar radiation on them. And the curious thing about the glacier algae is that you might think that preferentially they grow on south-facing slopes in the northern hemisphere, but in fact they preferentially grow on the north-facing slopes. So these glacier algae preferentially like to be out of the sun, rather than on the steep sides of these hummocks that are facing south. So they don't necessarily like too much light. But I think there might be another explanation, which I'll mention in a moment. We did a lot of sampling. Very crudely categorized the surface ice as having low, medium, and high algal compositions. Did a whole pile of incubation experiments, melted a whole lot of ice so that we could determine how much particulate is in it and what the ice algae content was. Now I'm slightly dyslexic, so I butcher the English language, and when it gets Latin, it becomes even worse for me, and I can never remember names so well. But the biodiversity of the glacier algae is quite restricted, so it's usually just 2 types of organism, and it's Ancylomenia, Mesotanium, and it's usually just these 2 species of algae. And I was asking the microbiologist, why just 2? And their line back is that they're the only 2 organisms that can grow there, and there's no other competition, but they just have the right characteristics to survive on the ice surface. Did a bunch of hyperspectral measurements, and you can see that relative to the white ice, light algal cover depresses the albedo, and the heavy algal cover takes the albedo, here reflection, down to reflectance, down to 0.2 and even down to 0.1. guys ran a bunch of drones, and I won't embarrass myself. I know relatively little about this, so I had a great time down in the smart lab. But Joe Cook, Andrew Tedstone produced DEMs. They undertook hyperspectral measurements of spot samples. Joe had, I will embarrass myself because I can never remember the wavelengths that he operated under. But he had and I've forgotten the name. He had sensors calibrated to different wavelengths, a little like we saw down in the smart lab, and he was able to use ratios of one wavelength to another to map what he thought were algal distributions over the snow cover. And I'll give you some references to his work and Andrew's work at the end of the talk. Now, what I'm much more comfortable with is the microbe and the particulate interactions, and this is work undertaken by Chris Williamson. And his brief was to try to understand what kick-started the ice algal blooms, and indeed where the ice algae first started growing. And for women in science, it was really quite interesting that the volunteers for the spring snow sampling, which is pretty hardcore, there were 3 women and just one guy. So the girls are hardcore on this project. What did he find? Well, he found that indeed the glacier algae are frozen into the surface ice over winter and quite happily survive at depths somewhere between 10, 20, 30 centimeters. And that in the spring, as the snowpack starts to decline and meltwater starts to rain down on the underlying ice, sufficient light gets through to warm up the ice algae and their little microenvironments in the ice. And the ice algae start growing in the ice underneath the snowpack. So we know that the ice algae can survive the winter in the ice, and the algal blooms start initiating within the ice before the snow line retreats. We wondered what was limiting the algal growth, and the usual thing to do is to add. You have a control, which is just ice melt with algae in it. You add nitrogen in the form of nitrate, phosphate, add N and P, incubate the little bottles, some illuminated under natural conditions, other with foil around them to block out the light. And so the light bottles should photosynthesize and produce oxygen. The dark bottles should respire and take oxygen out of the water. And you can gain an idea then of what the productivity was. Chris's particular interest is in the photophysiology of the ice algae, and what he was doing was looking for evidence of the difference in the electron transport rate, the efficiency of the glacier algae in dealing with the incoming solar radiation. And the kind of odd thing is that you might think an ice sheet surface hasn't got very much nutrient in it. Well, you add nutrient 5 times those of the ambient concentration in the snow and ice. And if anything, my reading as a chemist is you add nitrate, and actually that suppresses that suppresses photosynthesis and respiration. Phosphorus, a little bit the same. Chris's more statistical take on the data is that there's no difference between algae growing in just ice melt and algae fertilized with nitrate, phosphate, and a combination of both. So they don't seem to be nutrient limited. That didn't seem to make a lot of sense to us, and what we thought we would do in the following year is to add slightly different types of things to the incubations. And we noticed that most of the dissolved inorganic nitrogen in the snow and in the ice was in the form of ammonia rather than nitrate. So we added ammonia and nitrate and phosphate and all. And the key thing that we thought might be useful was particulates, because the glacier algae were always associated with particulates. And I think what this graph shows here is that somehow you place the algae in solution and illuminate it in the presence of particulates. And the ice algae, they grow much more comfortably than if they're left in ice melt with various different treatments. And the interesting thing I thought here is, the interesting thing here, I thought the cocktail of chemicals that works the best includes ammonia this time. So I thought potentially then the ice algae responded to both particulates and to ammonia over nitrate. So there's a need then, if you want to know about glacier algae growth, there's a need to understand what the particulates are in the surface layers. And here's where Janine McCutcheon comes in. And she's going to go to McMaster's, I think, in March of next year. She's a really bright woman, very good on particles and on the characterization of black and brown carbon particles. So, her work is very detailed, very meticulous. Even doing simple things like characterizing the types of particles that you find in aerosol samples and in ice melts takes a long time. What she found in the air samples were windblown dust, pollen, of course, and some black carbon. So, you know, there is black carbon being deposited on the ice sheet surface, but there's not enough to darken the ice sheet surface. What she did was to undertake an analysis of particulates in clean ice, snowfall, weathered snow that's wet and melted. Early season clean ice, late season clean ice, high algae biomass, dispersed cryoclonite ice, and you go then from white stuff to dark sooty stuff, and it's a log scale, and these are crazy units of nanograms per gram of water. And the thing to get then is that somehow, if you look at the particulate loading of the clean ice and the new snowfall, they don't contain as much particulate material as those of the dirty ice and even the early season relatively clean ice in which the glacier algae is starting to grow. When you melt these solutions, you know, I think that's the best illustration of how dirty they are. It looks like soot that you're filtering out the solution. when you look at the organisms by SEM, under SEM, you can see that there are cyanobacteria that seem to trap and bind the particulates together, and that you find in cryoconite. But so they say, and it's not published yet, but there are different types of cyanobacteria that grow up on the ice surface and that bind the particulates tightly together. And in this particulate mass, it contains mineral matter. It's about 5% organic carbon by weight, and about 95% by weight of minerals. But that's not, on a volume by volume basis, it would be much nearer 50-50. There are, a bunch of large minerals, and then scattered through the mineral surface are microorganisms, and there are viruses in the surface samples as well. And one thing that Janine's been finding is that the microbes like to bind to the particle surfaces, and it looks like they are biomining phosphate from the mineral surfaces. So they're keyed up to bind onto larger mineral surfaces, and it looks like they're biomining phosphorus from the mineral grains. She's done a bunch of analysis on the mineralogical components to the debris. 65% of it is quartz, albite, and andesite. They're kind of white-grey translucent minerals. There are some green bloom minerals, diopsite, actinolite. There's some red minerals, standardine, orthoclase, titanite, and there are some truly dark minerals, black minerals, hornblende, ensitite, augite. And the thing is, then, that if you're using absorption ratios, if you're using the red edge to distinguish glacier algae from background, there's always an interfering effect from these, the 7% of the red minerals, and you just have to accept that's the way it is, and it's very difficult to correct for. What do the soot particles look like? And this work is not published at the moment, so I'm skirting over it. But I just want to showcase that if you want to know about these sorts of things, then Janine is a good person to work with. There are black carbons from high temperature industrial emissions. There are brown carbons from forest fires, for example. And Janine has the skill base, knows the tools to use to be able to distinguish between the different types of carbon that you might find at the surface. Now, if you're interested in a summation of this, so Joe Cook has got a paper that we're trying to get published in the cryosphere, and what he's done is to use remote sensing imagery to some would say rather crudely deconvolute how much of the albedo is due to glacier algae and how much is to do with other light absorbing particulate material. And the kicker is that about 10% of the melt generated in the dark zone comes from the direct effect of dark glacier algae. Some parts of the heavily pigmented areas on the ice surface, they can contribute about 30. Glacier algae then would contribute about 30% of the meltwater. But overall, along the western margin of the ice sheet, about 10% of the melt is being generated directly from the glacier algae. There's another paper again in review, and it's the cryosphere. This time it's Andrew Tedstone. And what he's done then is to look at the other components of albedo reduction, and he's come up with the idea that it's the architecture of the weathering crust that's very important to determining just how much melt there is. And there's a dynamic between where the algae are, how they melt the surface, how much water the surface then retains. And Andrew's kind of best estimate is there's another 10% possibly of the melt that's generated indirectly by the glacier algae changing the physical structure of the weathering crust, and that is in terms of the ice crystal size and the water content of the ice surface. And you know, please feel free to check out what I've said. Never believe anything I say. You know, check me out in what the written word is. So, what about the future? Well, we've had a glimpse already of what the future might bring. This June, July, there were record hot waves. There was about 60% of the ice sheet, up to 60% of the ice sheet surface was melting. And the thing about the melt zone expansion, it goes towards the interior of the ice sheet, where the ice is flatter. Now, you might ask why the algae don't grow on the ice sheet, on the very margins of the ice sheet, and it's probably because the ice sheet margins are steeper, and the ice algae can get flushed out of the weathering crust as the melt season develops. But on the flatter surfaces of the ice sheet interior, where the water will pond for longer, it's possible that more and more favourable conditions for ice algae growth will occur in a future warming world, and what we intend to do with the Deep Purple project then is to look at the interaction between the glacier algal ice growth, the weathering crust evolution, and the particulate geochemistry and the biomining, because our hypothesis is that only if you know something about microbiology, particle biogeochemistry, physical glaciology, can you understand the three big effects that are controlling darkening. And so, you know, hopefully it won't be 20 years. Please invite me back before I retire in 6 years' time, because the Deep Purple project, what we're trying to do is to ascertain whether the dark zone will expand further and further into the ice sheet and further north in the ice sheet in a warming world. And the points that we make are that, you know, the Greenland Ice Sheet is one of the least studied of the Earth's biomes. We know a lot about one site, but we don't know so much about other places on the ice sheet surface. We still don't really understand the fundamental controls, even though we've been studying it for 3-4 years now. What controls the ice algal growth? And so therefore we can't understand what the future algal contributions to melting will be. So what we hope to do over the coming years is put together some conceptual biogeochemical models, which will involve glacier algae, particulates, and the architecture of the weathering crust. And from that, we will produce albedo parameterizations that can be used by proper ice sheet modelers. And I suppose our contribution to the field is that there's relatively few field observations around the ice sheet. There are relatively few people who are interested in the actual processes that occur at the ice sheet surface, and we're going to fill that gap, hopefully, over the next 4 or 5 years. So, this is 3 people, one looking very shocked and doesn't understand how you do selfies. Alex Anicio in Aarhus, Liana Benning at Potsdam, and myself as the old person on the way out. I'm going to be on a farewell tour. And hopefully I can come back and give you the results of what we found. 5 years' time, John, will you still be here? Yeah, okay. And yeah, I look very, very much forward to coming back then. So thank you all for listening, and I hope that you found it interesting.
Great. Well, thank you, Martyn. We have time for some questions. Maybe start off with one.
Yep.
Okay. As you'd expect the melting zone to expand over the Greenland ice sheet, how will the algae be transported to areas where they've formerly never colonized?
Well, thank you, John. And why didn't I tell you that we do find them in the? I should have said that Janine's been finding them in the aerosol sampling that she's done.
So they're blowing with the.
Some of them get blown off the surface in the late summer and autumn.
Right.
They're lofted, and then they're bashed out of the atmosphere by the snow. So you don't find so many of them in the snow, but there are some there.
Right.
And that will be sufficient to colonize, the right location.
Mm-hmm. Okay. Kevin.
Are there similar types of blooms happening in other places like Antarctica, or?
So not so much in Antarctica yet, although the peninsula is starting to warm. So there's relatively little surface melt in Antarctica. Much of the melt is by dust below the surface. Curiously, where there's enough dust, then you can generate subsurface melt. But were you to go to the Alps, I bet you I know in the Canadian Rockies there are ice algae. In the Himalayas, just everywhere, there's always wherever there's water, there's life, and you'll find glacier algae on glaciers worldwide. Do they have quite the impact on the surface albedo that you find in Greenland? Well, probably not, because the smaller valley glaciers have a lot of dust blown on them. But attached to that dust, I bet you'll find just everywhere you go, you'll find glacier algae growing, and they're very, very likely to be just 1 or 2 species.
Are there any downstream effects from algae-rich meltwater?
So not that we know of at the moment, but what they do is they convert inorganic nutrients, so nitrate, phosphate, into dissolved organic forms. So dissolved organic phosphorus, dissolved organic nitrogen. So they will maximize the potential of the surface to scavenge solute from whatever source it could, nutrient from whatever source it can get. The exudates from them produce dissolved organic matter. So these things are carbon factories. One way they deal with the high light intensities is they just produce goo, and that goo is very labile to downstream heterotrophs. Now, whether that's how big an impact it has downstream, then I don't know. But they certainly impact on the dissolved organic carbon content of the water, but they're not huge concentrations. So at the moment, not so much, is my guess. Oh, I held my heart just like I did 20 odd years ago.
Right. Martyn, what's the goo?
The goo as I'm getting older, I keep forgetting the name. It's EPS, so it's extracellular substance.
So it's goo.
Talking like
Yeah.
it's goo.
Lovely.
Yeah,
Okay.
I'm an old-style geochemist, and you think that, old-style geochemists think that microorganisms, they have fixed carbon, nitrogen, and phosphorus ratios, just like the phytoplankton that are in the ocean. And you think that to grow, or the plants in your pots in the house, they need a fixed amount of nitrogen and phosphorus to grow. But that's not the same with microbes in highly illuminated areas. So the way they deal with light stress is to fix more organic carbon. So they turn into organic carbon producing factories to dump off some of the excess light. And so over the growing season, their carbon to nitrogen to phosphorus ratio changes. So, and it's wacky that you would think that in the heavily populated dark areas that there must be more phosphorus and nitrogen, and that is true, but there's not so much there as you need, because the CNP ratios have all got higher and higher and higher. So it's a wacky environment where these, what you might otherwise think would be fixed carbon, nitrogen, phosphorus ratios, they're actually very plastic, and they change with the amount of light stress and with the amount of environmental stress that the organisms are experiencing. So that,
Hi, thank you. Is it possible to reproduce the mechanisms that you show with the darkening, the glacier algae, and the darkening of the albedo in a laboratorial environment?
Oh, well, that's a great question. And we would have said, up until 8-9 months ago, we would have said, we can't do it because you can't culture the organisms. So when we bring them back, just like you would bring back microbes from any other environment that you might want to sample, they don't grow particularly well in laboratory cultures. And what my colleague Alex Anicio has been able to do in Aarhus, he's found a way of making them grow in the lab. And then Chris Williamson, by serendipity, also found another way to make them grow. But they don't grow purple, they grow green. So we can grow them as green algae, but we can't grow them as purple algae at the moment. So it seems that you need the combination of ice and particulates and the particular lensing effects that are produced to turn them purple. So we made a big breakthrough in that we can now culture them, and we have the potential to do lab experimentation. And that's only on the timescale of the last 8 or 9 months. But at the moment, we can't turn them purple.
Martyn, many people in the room here are familiar with pink snow that grows in alpine snowpacks in the Rockies, sometimes on glaciated areas like in Glacier National Park. How does this algae relate to that? Because the albedo impacting properties of watermelon snow have been known for some time.
Oh no, it's the same sort of effect, but these glacier algae have a particular type of pigment called, and then Chris Williamson will crack himself up if he ever listens to this, because I can never say the word, purpulogalin. So these glacier algae have a specific type of pigment. It's not found in any other microorganism that we've been able to find to date, and it's a kind of it's a super sunblock. So I don't know if we could patent it, if we could make a paste and patent it, but it's a super sunblock, and it operates right the way across the solar spectrum.
Because that's what's happening in pink snow algae as well.
Yep.
They're that color to survive in a high UV environment. So,
Yeah.
Interesting. Do you get snow algae on top of these ice layers in Greenland?
So my colleague Liana Benning and her postdoc Steffi Lutz were kind of so upset that they can barely find an otherwise terrestrial snow algae. So you find them down the south,
Right.
but as you go further and further north, you don't find them. And I think that's because there's not enough organic matter for them. So
Okay.
back in our old snow ecology
Yeah,
days,
yeah.
Ron Hoham was always telling us, if you've got organic matter in the snowpack, you know, you've got wind-blown organic matter in the snowpack, then the snow algae are really happy. What they don't like is organic-free areas, as I crudely understand it.
Anyway, great. Want to thank you, Martyn. That was wonderful.
Thank you very much.
And yeah, I think we'll be retiring to Boffins to further discuss interaction of microorganisms in water.